Hello students, today we'll be studying the beta oxidation of fatty acids. Let's go through the introduction first. The oxidation of long-chain fatty acids to acetyl-CoA is a central energy yielding pathway in many organisms and tissues. In mammalian heart and liver, for example, it provides as much as 80% of the energetic needs under all physiological circumstances. Although the biological role of fatty acid oxidation differs from organism to organism, the mechanism is essentially the same. The main forms of fatty acid oxidations are termed alpha, beta, and omega, of which beta oxidation is the most common type. The repetitive four-step process called beta oxidation, by which fatty acids are converted into acetyl-CoA, is the main topic of this lecture. Long-chain fatty acids, combined as triacylglycerols, provide the long-term storage form of energy in the adipose tissues of animals. In addition, many plant seeds contain triacylglycerol stores. The properties that make triacylglycerols good storage compounds, however, present problems in their role as fuels. Because they are insoluble in water, ingested triacylglycerols must be emulsified before they can be digested by water-soluble enzymes in the intestine, and triacylglycerols absorbed in the intestine or mobilized from storage tissues must be carried in the blood bound to proteins that counteract their insolubility. To overcome the relative stability of the carbon-carbon bonds in a fatty acid, the carboxyl group at C1 is activated by attachment to coenzyme A, which allows the stepwise oxidation of the fatty acyl group at the C3 or beta position, and hence the name beta oxidation. The mechanism was originally proposed more than a century ago, that is in 1904, by Knoop. Let's look at the cellular site of beta oxidation. Earlier, it was considered that beta oxidation was confined to the mitochondria. Although animal mitochondria do contain all the enzymes necessary and are a major site for beta oxidation, other subcellular sites, such as the microbodies, are implicated. Peroxisomes or glyoxysomes together are often referred to as microbodies. They contain a primitive respiratory chain where energy released in the reduction of oxygen is lost as heat. Microbodies occur in all major groups of eukaryotes, including yeasts, protozoa, plants, and animals. The contribution of these organelles to total beta oxidation in a given tissue varies considerably. In animals, microbodies are particularly important in liver and kidney. In fact, in liver, it seems that mitochondria and microbodies collaborate in overall fatty acid oxidation. Thus, the microbodies oxidize long-chain fatty acids to medium-chain products, which are then transported to mitochondria for complete breakdown. In this way, very long-chain fatty acids, such as erucate, which are poor substrates for mitochondria, can be catabolized. Microbodies are important for the degradation of other lipid substrates, such as some xenobiotics and eicosanoids. In contrast, the glyoxysomes from germinating seeds are capable of the complete breakdown of fatty acids to acetyl-CoA. They also integrate this metabolism with the operation of the glyoxylate cycle, which allows plants, in contrast to animals, to synthesize sugars from acetyl-CoA. Leaf tissues also contain peroxisomes, and it is found that beta oxidation in leaves is always found in peroxisomes with significant activity in mitochondria in some circumstances. Next is activation and transport of fatty acids into the mitochondria for oxidation. The enzymes of fatty acid oxidation in animal cells are located in the mitochondrial matrix as demonstrated in 1948 by Eugene P. Kennedy and Albert Lehninger. The fatty acids with chain lengths of 12 or fewer carbons enter mitochondria without the help of membrane transporters. Those with 14 or more carbons, which constitute the majority of the free fatty acids obtained in the diet or released from adipose tissue, cannot pass directly through the mitochondrial membranes. They must first undergo the 3 enzymatic reactions of the Carnitine shuttle. The first reaction is catalyzed by a family of isozymes. Different isozymes specific for fatty acids having short, intermediate, or long carbon chains present in the outer mitochondrial membrane, the acyl-CoA synthetases. Thus, acyl-CoA synthetases catalyze the formation of a thioester linkage between the fatty acid carboxyl group and the thiol group of the coenzyme A to yield a fatty acyl-CoA, coupled to the cleavage of ATP to AMP and pyrophosphate. The reaction occurs in 2 steps and involves a fatty acyl adenylate intermediate. Fatty acyl-CoA esters formed at the cytosolic side of the outer mitochondrial membrane can be transported into the mitochondrion and oxidized to produce ATP, or they can be used in the cytosol to synthesize membrane lipids. Fatty acids destined for mitochondrial oxidation are transiently attached to a hydroxyl group of carnitine to form fatty acyl carnitine, the second reaction of the Carnitine shuttle. This transesterification is catalyzed by carnitine acyl transferase 1 in the outer membrane. Either the acyl-CoA passes through the outer membrane and is converted to carnitine ester in the intermembrane space, or the carnitine ester is formed on the cytosolic face of the outer membrane, then moved across the outer membrane to the intermembrane space. In either case, passage into the intermembrane space occurs through the large pores formed by the protein porin in the outer membrane. The fatty acyl carnitine ester then enters the matrix by facilitated diffusion through the acyl carnitine transporter of the inner mitochondrial membrane. In the third and the final step of the Carnitine shuttle, the fatty acyl group is enzymatically transferred from carnitine to intramitochondrial coenzyme A by carnitine acyl transferase 2. This isozyme, located on the inner face of the inner mitochondrial membrane, regenerates the fatty acyl-CoA and releases it along with the free carnitine into the matrix. Carnitine re-enters the intermembrane space via the acyl carnitine transporter. This 3-step process for transferring fatty acids into the mitochondrion, esterification to CoA, transesterification to carnitine followed by transport, and transesterification back to CoA, links 2 separate pools of coenzyme A and of fatty acyl-CoA, one in the cytosol and the other in the mitochondria. These pools have different functions. Coenzyme A in the mitochondrial matrix is largely used in oxidative degradation of pyruvate, fatty acids, and some amino acids, whereas cytosolic coenzyme A is used in the biosynthesis of fatty acids. Fatty acyl-CoA in the cytosolic pool can be used for membrane lipid synthesis or can be moved into the mitochondrial matrix for oxidation and ATP production. Conversion to the carnitine ester commits the fatty acyl moiety to the oxidative fate. The carnitine-mediated entry process is the rate-limiting step for the oxidation of fatty acids in mitochondria and, as will be discussed later, is a regulation point. Once inside the mitochondria, the fatty acyl-CoA is acted upon by a set of enzymes in the matrix. Next, now let's look at the mitochondrial beta-oxidation. Mitochondrial oxidation of fatty acids takes place in 3 stages. In the first stage, which is beta-oxidation, fatty acids undergo oxidative removal of successive 2 carbon units in the form of acetyl-CoA, starting from the carboxyl end of the fatty acyl chain. In the second stage of fatty acid oxidation, the acetyl groups of acetyl-CoA are oxidized to carbon dioxide in the citric acid cycle, which also takes place in the mitochondrial matrix. In the third stage, the NADH and the FADH2 produced in the first 2 stages donate electrons to the mitochondrial respiratory chain, through which the electrons pass the oxygen with concomitant phosphorylation of ADP to ATP. The energy released by fatty acid oxidation is thus conserved as ATP. We now take a closer look at the first step of fatty acid oxidation, beginning with the simple case of a saturated fatty acyl chain with an even number of carbons, then turning to the slightly more complicated cases of unsaturated and odd-numbered chains. Let's come to the enzymes of the mitochondrial beta-oxidation. The reactions of beta-oxidation are shown in the figure and essentially involve 4 enzymes working in sequence, that is acyl-CoA dehydrogenase, enol hydratase, a second dehydrogenase which is 3-hydroxyacyl-CoA dehydrogenase, and a thiolase, which results in the cleavage of 2 carbons at a time from the acyl chain. So 4 reactions remove each acyl-CoA unit from the carboxyl end of a saturated fatty acyl-CoA. Number 1, dehydrogenation at carbon 2 and carbon 3 by FAD-linked acyl-CoA dehydrogenases. Number 2, hydration of the resulting trans-2 enol-CoA by enol-CoA hydratase. And third, dehydrogenation of the resulting L-beta-hydroxyacyl-CoA by NAD-linked beta-hydroxyacyl-CoA dehydrogenase. And 4, CoA requiring cleavage of the resulting beta-ketoacyl-CoA by acyl-CoA acetyl transferase or thiolase to form acetyl-CoA and a fatty acyl-CoA shortened by 2 carbons. The shortened fatty acyl-CoA then re-enters the sequence again. The 2-carbon product acetyl-CoA is then used by, for example, the TCA cycle to yield energy, and the acyl-CoA, 2 carbons shorter, is recycled. Each of the 4 enzymes involved are present as isoforms that have varying chain length specificities. Thus, for the efficient degradation of typical fatty acid substrates, the different isoforms must cooperate. 3 of the enzymes, hydratase, 3-hydroxyacyl dehydrogenase, and thiolase, have their long chain isoforms present as a trifunctional complex on the inner mitochondrial membrane. All the other isoforms of the component enzymes are soluble, but there is evidence of metabolite channeling between the individual enzymes. This improves the efficiency of beta-oxidation and prevents intermediates building up, which could be inhibitory and also lead to a greater requirement for scarce CoA. As a generalization, it is thought that long-chain acids are oxidized by the membrane-located enzymes, while medium-chain substrates use the matrix enzymes. Other fatty acids containing branched chains, double bonds, and an odd number of carbon atoms can also be oxidized. From acids of long chain length, one of the products is propionic acid, and the ability of the organism to oxidize such fatty acids is governed by its ability to oxidize propionate. For example, liver is equipped to oxidize propionate and therefore deals with odd chain acids quite easily. Heart, on the other hand, cannot perform propionate oxidation, and the degradation of odd chain acids grinds to a halt. The end product of propionate oxidation, succinyl-CoA, arises by a mechanism involving the B12 coenzyme and the biotin-containing propionyl-CoA carboxylase. Similarly, branched chain fatty acids with an even number of carbon atoms may eventually yield propionate, while the oxidation of odd-numbered branched chain acids proceeds by a different route involving 3-hydroxy 3-methylgluteryl-CoA or HMG-CoA. Most natural fatty acids are unsaturated. A typical unsaturated fatty acids might be linoleic acid, and at first, beta oxidation proceeds normally. But then a cis double bond is encountered at the wrong position, which is position 3. An isomerase exists to convert the cis-3 compound into the necessary trans-2 fatty acyl-CoA. This isomerase will also act on trans-3 substrates, though at lower rates. Once over this obstacle, beta oxidation can again continue to eliminate a further 2 carbons, and then dehydrogenation produces a 2-trans 4-cis decadenoyl-CoA from linoleoyl-CoA. The discovery of a 2,4-dienoyl-CoA reductase showed that this enzyme could use NADPH to yield 3-trans decanoyl-CoA as its product. The enoyl-CoA isomerase then moves a double bond to position 2, and beta oxidation can proceed again as normal. Next, regulation of mitochondrial beta oxidation. The overall rate of beta oxidation may be controlled by a number of well-known mechanisms. Number 1, availability of fatty acids. There are 2 aspects to this availability. First, fatty acids have to be made available by hydrolysis of lipid stores or from the diet. Second, as discussed, the entry of fatty acids in mitochondria is under careful regulation through the activity of carnitine acyltransferase 1. Second, the rate of utilization of beta oxidation products, which in turn can either lead to specific inhibition of a particular enzymes or to feedback inhibition of the whole process. The concentration of unesterified free fatty acids in plasma is controlled by glucagon, which stimulates, and insulin, which inhibits the breakdown of triacylglycerols in adipose tissue stores. Once the fatty acids enter cells, they can be degraded to acetyl-CoA or used for lipid synthesis. The relative rates of these 2 pathways depend on the nutritional state of the animal, particularly on the availability of carbohydrate. In muscle, the rate of beta oxidation is usually dependent on both the free fatty acid concentration in the plasma as well as the energy demand of the tissue. A reduction in the energy demand by muscle will lead to a buildup of NADH and acetyl-CoA. Increased NADH-NAD+ ratios lead to inhibition of the mitochondrial TCA cycle and increase further the acetyl-CoA or CoAsh ratio. Kinetic studies with purified enzymes show that the major sites of beta oxidation inhibition are 3-hydroxyacyl-CoA dehydrogenase, which is inhibited by NADH, and 3-ketoacyl-CoA thiolase, which is inhibited by acetyl-CoA. In the liver, because of the interaction of lipid, carbohydrate, and ketone body metabolism, the situation is more complex. In the fed state, when carbohydrate, which is in the form of glucose, is being converted to fatty acids, the level of malonyl-CoA is raised. Malonyl-CoA is a reversible inhibitor of carnitine acyltransferase 1, and therefore reduced entry of acyl groups into the mitochondria for oxidation. In the fasting state, lowered malonyl-CoA allows carnitine acyltransferase 1 to function at high rates, and this stimulates beta oxidation and ketogenesis. In the fasted condition, a high glucagon-insulin ratio elevates the cellular cyclic AMP, thus allowing short-term inhibition of acetyl-CoA carboxylase by phosphorylation. Reduction of the glucagon-insulin ratio on feeding reverses this effect. Thus, fatty acid synthesis and degradation are co-regulated by liver hormonal levels. Now let's come to the conclusion. Number 1. Beta oxidation is the repetitive 4-step process by which fatty acids are converted into acetyl-CoA. Second, fatty acids are activated at the lower mitochondrial membrane by conversion to fatty acyl-CoA thioesters. Fatty acyl-CoA to be oxidized enters the mitochondria in 3 steps via the carnitine shuttle. And number 3, the reactions of beta oxidation of fatty acids involve 4 enzymes working in sequence. They are acyl-CoA dehydrogenase, enol hydratase, 3-hydroxyacyl-CoA dehydrogenase, and a thiolase, which results in the cleavage of 2 carbons at a time from the acyl chain. Number 4, although animal mitochondria are a major site for beta oxidation of fatty acids, it also occurs in microbodies like peroxisomes or glyoxysomes. The availability of fatty acids mainly through the activity of carnitine acyltransferase 1 and the rate of utilization of beta oxidation products are the main factors regulating mitochondrial beta oxidation.